And let's begin the meeting. I'm probably not speaking loud enough for most of you. Let's share the sound. There we go. All right, hopefully you can see the screen. We're going to be finishing chapter 1 today, and I hope to get started with chapter 2, chemical principles. Please note that you have a practice quiz due on Wednesday, and there's actually 2 of them. One is a practice essay questions, so a quiz on essay questions, and that one you just need to open up any browser and answer the questions by the deadline, which I think is 11:59 p.m. on Wednesday. The other practice is a multiple choice timed quiz that once you begin, you have, oh gosh, a little over 1 minute for each question. And I think if there's 10 questions, you're given 10 minutes, plus you have 2 or 3 extra minutes. And so both of these quizzes are to get you to practice taking my quizzes. And there are 2 quizzes. There's an essay question or short answer question, as well as the multiple choice. The multiple choice quizzes must be taken by Respondus Lockdown Browser, and you must download the Respondus Lockdown Browser before you can take the quiz. It doesn't take too long to download the Respondus Lockdown Browser, but if your computer is not very big and powerful, meaning if it's a little slow, then when you're taking the quiz in Lockdown Browser, before you begin the quiz, you should shut down all the programs you don't need. Is everyone clear on that? Like I am running Microsoft Word to show you the syllabus. I've got 2 Zoom windows open here. I don't know if you can see that. I've got PowerPoint open, and then the what is that called? The button to show you where the heck I am on the in the folders. And what you want to do is shut everything down except for the Lockdown Browser, and then just have that running. If your computer is slow and, I don't know, not very powerful. Any questions about all of that? If you don't do that and your computer is slow, then you might get a delay in taking the quiz, and that'll eat up your time. So because it's a time quiz, you don't want that delay, and I'm suggesting you shut down all the programs you don't need. Obviously, you do need Lockdown Browser, Respondus Lockdown Browser. Any questions about that? These are both practice quizzes. You will get 1 extra credit point for each quiz if you take them, and you have correct answers. On the multiple choice quiz, I think there's about 10 questions, so you'll get 0.1 credit, extra credit per question. I don't remember how many questions there are. Any questions about that? Another reason for taking the practice quizzes is it'll force you to get the Respondus Lockdown Browser working on your system, because every once in a while students have a little bit of trouble getting it to run on their system. And if you're one of the ones where it gives you trouble, and like I said, it's usually not 1 student a term, it's usually like 1 student every 3 terms, contact the Computer Help Services. I don't remember what Clark calls that. The computer IT people, and they're an expert at getting the Respondus Lockdown Browser to work on your system. And usually when it doesn't work, it does tell you what you need to do to get it to work. And if you're computer savvy, you can do it yourself. But most people aren't terribly computer savvy, and so the IT services will help you get it running if you have trouble. And hopefully no one will have trouble this term. Any questions about that? All right, so that's what we're doing this week in the lab, and hopefully we'll get started with chapter 2. All right, so I believe I talked to you about the first observations that were done in microbiology by Antony van Leeuwenhoek. Is that correct? I'm not hearing anything, so I'm going to assume that is correct.
Yes.
Okay. Let's move on to the next slide. And that marked the beginning of microbiology, the observations of Antony van Leeuwenhoek. And I'm not Dutch, so I could be mispronouncing his name. Whoops, I thought I moved on. He built his own microscopes and ground his own lenses. And he was the best microscope builder and lens grinder of his day. He was so good at grinding lenses that he saw these cells here, which if you look at those and you know bacteria, you'll say, yeah, those are bacteria. These are rods. That's a bent rod. Those are cocci, and that's a spirillum. And then he's showing how the cell moved, swimming in a liquid. And these cells were so small that they were not seen for about a century later by other people. And it's just because he built the best microscopes and ground the best lenses. In his day, you had to have somebody grind your lenses, and he hand ground them. And why his microscopes were much better than anybody else's is, if you look at this, this is not a compound microscope. It only has one lens for magnifying. So really what it was is a magnifying glass, a super duper magnifying glass, and he got the best clarity from his microscopes. And he was able to see bacteria, cells so small that they weren't seen by other microbiologists for almost a century later. And what happened is, over time, they started building the microscopes better and better, and eventually they built them to the point where they had enough focus and clarity to actually see the bacteria. Any question about any of that? All right. The next thing, I'm not going to talk much about it, is Rudolf Virchow wrote up the theory of biogenesis, that all living cells arise only from pre-existing living cells. What's more important in the history of microbiology was Louis Pasteur. He finally disproved the theory of spontaneous generation, and therefore confirming the theory of biogenesis, that all living cells derive from other living cells. He demonstrated that microorganisms are present in the air, and that they can fall into things and then grow, and that microbes can be destroyed by heat. And therefore he was able to sterilize as media by heat, and then if you cap it, you could have sterile media. And then he also made the ingenious swan-necked flask and did an experiment which disproved spontaneous generation. What he did was he poured beef broth into a flask like this with a long neck glass, and then heated the glass and bent it into the swan neck. Because he heated it, this media was then sterile. And then when the microbes came down from the air, they could fall into the open glass swan neck flask, but the microbes would settle right there because of gravity. And because this flask was open to the air, the vital forces that were important for creating spontaneous generation could get into the flask. But the microbes did not. And so he was able to show that spontaneous generation of microorganisms did not occur. Any questions about any of that? If not, let's move on. Well, with that work, which, like I said, early microbiology was largely concerned with, could microbes, microbiology organisms, spontaneously generate or not? And then Louis Pasteur disproved it. Well, after that work, there came an explosion of data or information in microbiology. So much so that it's called the Golden Age of Microbiology. From about 1857 to 1914, you don't need to know the dates. It's just that during this time, a whole lot of things about microbiology were discovered. One of the big ones was the relationship that came out of Pasteur's work was that germs, microbes, could cause disease. And there was a relationship between microbes and organisms, and people suspected that microbes might cause disease. And that that theory developed over time. And it's called the germ theory of disease. Extremely important. Out of that came the concept of immunity, that we have an immune system that can protect us from germs that cause disease. And then out of both of those came the concept that we can make drugs which can work against meaning they work against microbes, antimicrobial drugs. And the best ones of these are antibiotics, and we'll talk about that shortly. So early on, Pasteur developed the basic idea of aseptic technique. You sterilize the media, you then cap it or cover it with the sterile something, and then you can get something to grow in it, and it'll be a pure culture. He did come up with the concept of a pure culture, and we'll talk about that, I think, in the lab. On top of Pasteur's aseptic technique, Louis Pasteur. So Dr. Koch developed the technique for streaking for isolation on a solid media, and what he did was he made it very easy to work with the pure culture. You can get a pure culture just by streaking the bacteria or the microbes on a solid media and getting isolated colonies. And if you do it 2 times, meaning you streak your culture through 2 isolated colonies, the chance of getting a pure culture is very high. And so that's why the streaking for isolation in solid media was very important for microbiology. On top of that, Hess, who worked in Dr. Koch's lab, developed nutrient agar, a new solid medium for growing mini microbes, and at the time it was the best media for doing that, and it was very simple and easy to make, as well as that it grew a lot of microbes. About the same time, this is when Sergei Winogradsky developed the concept that microorganisms are involved in the biogeochemical cycles, and we talked about how nitrogen is moving around on the Earth's ecosystem, and it's because the microorganisms are changing one form of nitrogen to another, allowing nitrogen to recycle on the planet and move around on the planet. Well, the same thing was shown for the sulfur cycle, that sulfur moves around on the planet because of microbes. All right, any question about any of these? Out of this work, Pasteur, so Louis Pasteur is very famous in early microbiology, showed that microbes are responsible for fermentation. He got into this research because the beer and the wine industry were having their cultures, their beer and their wine going bad, and they asked Louis Pasteur to find out what was going on. And Louis Pasteur showed that microbes are responsible for fermentation, that microorganisms convert the sugar in the liquids that they make into beer and wine, and then convert that sugar into alcohol, which makes the beer and the wine. And then he further showed that other microorganisms growing in the beer and the wine are responsible for spoilage of the beer and the wine. And what happened was that bacteria use the alcohol, metabolize it, and produce acetic acid, which would spoil the beer and the wine because it ferments the alcohol into vinegar or acetic acid. Any question about any of that? And if you've ever drank an old bottle of open wine, you notice that it's very tart, very sour. And that's because some of the alcohol has been converted into acetic acid. And that was spoiling the beer and the wine.
Quick question.
Uh-huh.
A quick question.
Uh-huh.
So why, what is the process of why the wine or the beer turn into vinegar? I mean, is that because attack another cell or?
Excuse me a minute here. I got to blow my nose. Go ahead and ask.
Sorry. It's funny, you know, because my family in Spain come from, you know, they make cherry wine. So I know the process, you know, but kind of curious about how the wine or the beer converts from beer or wine into vinegar.
When you take grape juice and you ferment it, you will get wine. Other organisms can ferment the alcohol in the wine into vinegar, and that is actually how they make wine vinegar. Okay, so it's usually two different organisms, one that'll ferment the grape juice into the wine, and then another organism that'll ferment the wine into the acetic acid.
Which is why when you're brewing beer or making wine, you want to maintain everything in the one cask and not let any excess yeast get into your batch.
Yeah, it's not yeast. It's usually bacteria that spoil the alcohol.
Oh, gotcha.
But
Okay.
if you can do it, usually the vintners usually don't do it sterile. Okay? Like they put preservatives in wine, and that's to prevent the bacteria from growing in it. But like I said, there's other things you can do. And let's move on to what can you do. Louis Pasteur came up with a very easy process of keeping the beer and the wine safe. And that is early on when the alcohol is made, you can pasteurize the beer and the wine, apply a high heat for a short period of time, which does not evaporate most of the alcohol. And then that killed off the bacteria that caused the spoilage of the beer and the wine. Now, I'm sure you've all here heard of pasteurization, although it's more common in dairy products. But Louis Pasteur invented it for keeping beer and wine I don't know, preserved. And I don't think anyone uses it anymore to, I don't think anyone pasteurizes wine, although they still pasteurize beer. I think the reason why they don't pasteurize wine is the pasteurization does evaporate some of the alcohol, and the vintners don't want that. In beer, it's not a big deal because beer has less alcohol than wine. Anyways, pasteurization, the application of high heat for a short period of time, is very common and used for a wide variety of foods. It does not sterilize the food. Like pasteurized milk does have a shelf life. And that's because it stops the spoilaging bacteria, but it does not kill all of the bacteria in the milk. And so after a certain amount of time, the bacteria that are still alive after pasteurization can spoil the milk. But pasteurized milk lasts longer than unpasteurized milk. Any questions about any of that? All right, let's move on. So with Louis Pasteur's work on fermentation, the realization that microbes can affect an organic material and then cause it to go bad, people were wondering, could microbes have a similar relationship with plants and animals? Might microorganisms cause disease? There is not one person who came up with the germ theory of disease. It was developed over time by many people. One of them, oops, sorry about that. One of them was Louis Pasteur, but many people contributed to the germ theory of disease. So not one person came up to it. And it was an important theory that microorganisms could cause disease, because obviously they do, but it differed from the original belief. Originally, people, like the medical profession, believed that poisonous vapors were responsible for causing infectious diseases. Like they believed that if you walk through a swamp, you could get cholera from breathing in the poisonous vapors of the swamp. Of course, we now know that cholera is located in the swamp, and then people who walked through the swamp breathed in some of the organism causing cholera. Okay? Any questions about any of that? If not, let's move on. I'm going to skip the germ theory of disease to people who are important in developing the germ theory of disease. You can read about it. I'm not going to quiz you on that. But the germ theory of disease was proved by Robert Koch. Dr. Koch did not propose the germ theory of disease. He proved it. What he did was he took the bacterium Bacillus anthracis and studied it and showed that it could cause the disease anthracis. He then provided the experimental steps required to show that Bacillus anthracis caused the disease anthrax, and said that these are the steps you need to show that any given microorganism causes a specific disease. And these steps are known as Koch's postulates. With that, he proved the germ theory of disease. A given organism causes a given disease, anthrax. And he published the steps he used, known as Koch's postulates, 2 years later. You don't need to know the dates. Almost everything I give you. Okay? Any question about any of that? There's only one date I asked you to remember, and that was the date when we first saw microorganisms in the fossil record. That's the only date I've asked you to remember. All right. Something that happened long before microbiology was started was the concept of vaccination. The first vaccine was developed by Edward Jenner in 1796, and that was at least 50 years before the majority of the things that gave rise to microbiology began. He made the first vaccine against the disease smallpox. What he did was he inoculated people, or a person, with the cowpox virus. And then he noted that the person was protected against the disease smallpox. He and everyone of his day did not understand why his vaccine worked. We did not know why the cowpox virus protected people against smallpox. And it was not understood until Louis Pasteur experimented with it, and he went on and showed what was going on and why vaccines work. What he showed was that a vaccine works because the agent that we use as the vaccine is related to the agent that we're vaccinating against. And so when our body makes a response to the cowpox virus, it also sort of accidentally made a response to the smallpox virus. And that's why the cowpox virus worked as a vaccine against smallpox. Any question about any of that? By the way, I can tell you that with coronavirus, COVID-19, it is believed that if you get a cold that's caused by coronavirus, within a few years before you get COVID-19, that your body will mount an immune response against COVID-19 because it has mounted an immune response against the coronavirus causing the human colds. Any question about any of that? If you don't know, coronaviruses are known to humans, to man, and most of them cause human colds. However, one of them causes COVID-19. Any question about that? And some of the viruses that cause human colds, maybe all of them, I don't know. Some of them
I got a question.
protection against COVID-19. Somebody was saying something?
I get a question. I do work in, in the call center at the VA.
Uh-huh.
And anyway, I have to schedule all the veterans to get the vaccine. So I'm very curious. So is that true that, these vaccines will get at messenger
Messenger
RNA?
RNA, yes.
Mm-hmm.
So what's your question?
My question will be, will that alter the immune response in the patient, sir?
Yeah, that makes the patient respond. Okay?
You think
All right.
they will alter the immune system? I mean, they will...
No, this vaccine has been shown to be very effective. What it does is it puts messenger RNA from COVID-19 into the patient, so there are no viruses, and then your own cells make the proteins of COVID-19 and then attack those proteins, and then when you get the virus, your immune system is attacking the proteins on the virus. Okay? So that's a very good question, but hopefully that's a short enough answer because I want to move forward. All right. Vaccination is actually a term derived from the word vacca, which is Latin for cow. So vaccination comes from the concept that the first vaccine was from the cowpox virus, a virus that attacked cows, and people got it too, okay? The protection of a vaccine is called immunity, and we'll talk a little bit more about that later in the term. All right? From all of these concepts came the birth of modern chemotherapy. Chemotherapy, strictly speaking, is a treatment of a disease with chemicals. Now, chemotherapy is used commonly nowadays to mean an anti-cancer therapy, which cancer is a disease which is treated with a chemical, so that works. But chemotherapy in the broader use is any treatment of any disease with a chemical. But most people, when they have a bacterial infection, we don't say, oh, it's time for me to take my chemotherapeutic drug to treat the bacterial infection. We say it's time for us to take our antibiotic. Okay? Chemotherapeutic agents against infectious diseases can be synthetic drugs. These are ones that are man-made, or antibiotics, which, strictly speaking, by the textbook definition, an antibiotic is a chemical that's produced by a living microorganism that inhibits or kills other microbes. That's strictly speaking the textbook definition of an antibiotic. But by that definition of an antibiotic, penicillin G is an antibiotic, but carbacillin, if I'm saying that correctly, and I think even ampicillin are not antibiotics, because with ampicillin, it's true a microorganism makes it, but then a human modifies it. And then carboxylin is totally a synthetic drug, because humans totally make that one, carboxylin. There's no microorganism that makes it. And most people would say, well, that's crazy, because these are drugs in the same family. And in common usage, the term antibiotic means any drug that is effective against a bacterial agent. Like carboxylin is considered by most people in common usage to be an antibiotic. And I will use the term antibiotic as it is used in common usage, because that is the way most clinics and most doctors use the term. All right? Any question about that? Now, the first chemotherapeutic agent used to treat an infectious disease was actually quinine that was found in the bark of the cinchona tree. You don't need to know that for this slide. And it was the first drug successfully used to treat a disease, which was malaria. That was the first drug ever used to treat a disease. Paul Ehrlich then went on to look for drugs to treat diseases, and he found the first drug to treat a disease, syphilis, in 1910. He called it a magic bullet to kill disease. And a patient could take his treatment and then be cured of syphilis as early as 1910. Now, the thing about his drug, Salvarsan, is it was a synthetic arsenic drug. And as you can imagine, an arsenic drug is a little bit toxic to people, but it could be used at the proper dosage to treat syphilis and not kill the patient. Obviously, if you use too much of the arsenic drug, it will kill the patient. Any question about that? So Paul Ehrlich found the first drug to treat a disease in 1910. The next drug that was found was by Alexander Fleming, who discovered the first antibiotic. What he did was he streaked bacteria out on a plate, and he was a poor microbiologist, and he got contamination of his plate. And then he left the plate on the counter and went away for the weekend, and then came back and then looked at the plate he streaked out, and sure enough he had 2 growth on it. The bacteria he streaked, and then a contaminant, a penicillium mold. But he noticed around the penicillium mold, as you can see, there's a region around it that has no growth. And he correctly concluded that something was coming out of this colony of mold, preventing the growth of the bacteria. And he called that an antibiotic. And penicillin, it's actually penicillin G, was the first antibiotic discovered that came out of the penicillium mold. Any questions about any of that? And Alexander Fleming did show that penicillin G did kill the bacteria Staphylococcus aureus. Unfortunately, Alexander Fleming was never able to purify penicillin G. And so it wasn't until the 1940s that anyone ever mass-produced it and clinically purified it so that it could be clinically used. So even though it was the first antibiotic discovered, it was actually the second antibiotic that was tested in the clinic. The first antibiotic that was tested in the clinic was a sulfur drug, and it was tested in 1935. It was man-made, and so it was pretty easy to get it pure. And so sulfur drugs were the first drugs that were clinically used, but penicillin came out as the second antibiotic and was used in World War II. Any question about any of that? If not, let's move on to the concepts of microbes and disease. Bacteria are once classified as plants, hence you'll see the term flora used to describe microbes. And sometimes I say microflora, and I apologize. I was instructed a long time ago when microflora was used to describe bacteria and other microorganisms, and now that has been replaced by the term microbiota. The normal microbiota are the microbes that are normally present in and on a human body. And all of us have a normal microbiota. And if you ever hear me use the word microflora, you can correct me and say, don't you mean microbiota? And I'll say, yes, thank you for the correction. Hopefully I won't do that, but if I do, I'm just warning you that in my day, microbiota, that term wasn't even invented yet, okay? So microbiota is the organisms in and on a person. And animals and plants host a symbiotic microbial community, very large numbers of cells that are part of our normal microbiota. And they're central to the animal and the plant's welfare. The normal microbiota are usually found on and in, well, on the skin, in the oral nasal cavities, the respiratory tract, the digestive tract, and the urethra. They are not normally found internal to the body, like in the blood, the internal organs, or the tissues. The normal microbiota are important in preventing the growth of pathogens. The normal microbiota can compete for resources and space, keeping the pathogen from growing on a person and causing disease. That is why one salmonella that you consume will not make you sick. You have to consume, I don't know, at least several hundreds. Salmonella is one that tends to make us sick, but like various types of E. coli that make us sick, we have to have a large number of them, maybe 100,000 of them, before a human will become sick. And that's because of our normal microbiota competing for the pathogen that would make us sick. Some of our normal microbiota secrete toxins that actually inhibit the growth of the pathogen. And we'll talk a little bit more about that later in the term. Some of the normal microbiota are important to us because they provide us with nutrients and growth factors. For example, humans, a large amount of folic acid and vitamin K is made for us by our normal microbiota. So if you're not taking vitamin K, at least you get some from your normal microbiota. You don't actually get 100%, but I think humans normally get about 33% of their vitamin K from their normal microbiota. And some of the vitamin K is in your diet, and others are supplements. I think green leafy vegetables are high in vitamin K, but we're not going to get into that. The normal microbiota is also important in helping you digest food. E. coli helps us digest food. Any question about any of that? Excuse me. The normal microbiota do not normally cause disease. In a healthy individual, the normal microbiota does not cause disease. However, the normal microbiota can cause disease in certain circumstances. Let me just say that if a patient has a weakened immune system, like AIDS patients, the normal microbiota may cause disease. Any question about that? I'm sure some of you know that you've had a bloom infection, like when you took antibiotics. What happens is not a bloom infection, a yeast infection, sorry, when the yeast bloom is what I meant to say. When you take antibiotics, it kills off the normal microbiota, and then the yeast can grow, causing a yeast infection. And you can get bacterial infection from taking antibiotics as well. All right, any question about any of that? Infectious disease results when a microbe gets on a patient, overcomes the host resistance, does damage, and then causes disease. So infectious diseases are caused by pathogenic microbes. All infectious diseases are caused by pathogenic microbes. A pathogen, if you remember, is just a microbe that causes disease. An emerging infectious disease is a new or changing disease that are increasing or have the potential to increase in incidence. So, right now, can anyone tell me an emerging infectious disease?
The coronavirus 19, COVID-19B.
Yeah, COVID-19. Are you talking about a specific strain?
Yeah, there's a new one, the 19B
All right.
or something like that.
All right, that's true. However, in reality, any COVID-19 is an emerging infectious disease. Because they're new, humans have seen them for a little more than a year, and they are increasing in the human population. Okay, and obviously the new strain is increasing even more rapidly. So that is a new and emerging infectious disease. Okay, any disease that has the potential to increase in incidence is also considered an emerging infectious disease. I will talk a little bit more about it later, but there was concern that bird flu could be an emerging infectious disease. It has the potential to increase in incidence. It first was noticed, I don't know, 10 or 15 years ago, that birds get flu, and then every once in a while they transmit it to people. And I think what happened was a large number of people in the chicken industry got bird flu, and then they were afraid that it might spread among humans, and so they were afraid it would become an emerging infectious disease. That never happened, fortunately, but you could say that it has the potential to increase in incidence in the future, especially if it were to mutate. Any questions about emerging infectious diseases? All right, so I want you to know the term emerging infectious diseases. You should know that coronavirus is an emerging infectious disease. They're new or changing diseases that are increasing, or they have a potential to increase in the near future. They can be caused by any type of microbe, viruses, bacteria, fungi, protozoa, prions, and helminths, which, remember, that's not exactly a microbe. And I've got a large number of emerging infectious diseases on here, but I'm not going to test you on emerging infectious diseases other than knowing what the term is. And I suppose you should know coronavirus is an emerging infectious disease. All right, any question about any of that?
I have a question.
Uh-huh.
Is MRSA a potential EID?
Yes. MRSA could be considered an emerging infectious disease. In reality, it's been decreasing because of the people aren't coming into contact with each other, but there was concern that MRSA would be spreading in the schools and in the nursing homes. So at least for a while it was an emerging infectious disease, only because of coronavirus has it been decreasing. But most people would probably consider it an emerging infectious disease. Okay? Any questions about any of that? If not, we're going to end this lesson and move on to Chapter 2. So if you have a question, ask it now.
Are you going to be sharing your slides for PowerPoint at all?
Yes, remind me. You should remind me in the lab because I won't have enough time after the lesson. Remind me in the lab and I'll post the PowerPoint slides.
Okay.
Okay, because I finished lesson 1 now. I think I want to save that. Sorry, this takes a while. Let's go on to Chapter 2. Do note that the notes are up. The notes do cover the same material that I lecture on. However, the notes won't show you it in a slide. It'll have it in sentence formation, or an outline formation. Sentence, but outline. All right. Chapter 2, Chemical Principles. Please know the major goals, not the only goals, meaning the major goals, not all of the goals. To see all of the goals, you should look at the objectives. And then a rough outline. Know the terms, know what is an organic chemical compound and what's an inorganic chemical compound. Know what are hydrocarbons. Know what polar molecules are and nonpolar molecules. Know what a hydrogen bond is. I don't think we go into the other types of bonds, but you should know what a different type of bonds are, like a covalent bond and an ionic bond. You should know what an acid is, what a base is, and know what pH is. You should be able to say, with respect to the major biological molecules, there are 4 of them, be able to describe the relationship and characteristics of the monomers and the polymers. And what are the polymers for the 4 major biological molecules? 3 of them are very easy. You can find it on anything you eat, like a cereal box. What are the 4 major biological molecules? Come on, somebody's got to give it to me.
Nitrogen, carbon, oxygen, hydrogen.
That's a good guess, but no, those aren't biological molecules. Those are elements. Tell me a biological molecule, you'll find it like on a cereal box. What do you find on a cereal box?
Lipids, protein.
Lipids and proteins,
Carbohydrates.
carbohydrates, and the
Nucleic
4th one
acid,
isn't on the cereal box.
nucleic acids.
Nucleic acids, yes, those are the 4 major biological molecules. They're the molecules of life. They're called that because you only find these molecules in things that are alive or were once alive. So if you find it in a rock, it's probably from a fossil, okay? Something that was once alive. But you will not find these 4 major biological molecules like on a rock from the moon. Be able to describe the composition, the structure, and the function of the 4 major biological molecules, and be able to give examples of each. All right, any questions about what we're going to be looking at, what the major goals are? If not, let's take a look at the scale of things. Let me see if I can blow that up a little bit. Move this over. Actually, I can do it easier from this way. Ta-da. You should know the scale of things. I want you to know the relative order. Like a human is bigger than a chicken egg, which is bigger than a frog egg, which is bigger than the eukaryotic cells, the average eukaryotic cell, which is bigger than the prokaryotic cell, which is bigger than a virus, which is bigger than a large molecule, like a protein, which is bigger than a small molecule, like water, which is bigger than a atom. So you should know the relative order of things. And let me point out there's an error in this chart. You see 1 micrometer here and 1 micrometer here. Right there, that should be 1 millimeter. Okay? You do not need to memorize the sizes of things and the numbers, but you should know the relative order of things. Any question about that? Okay, what are organic compounds? Can anyone tell me? What's an
Yeah.
organic compound?
Something that always contains carbon
Yes?
and hydrogen.
Yes, it has. An organic compound always has carbon and hydrogen. Okay, we'll go with that. That's actually pretty good. Typically, they are complex molecules, organic compounds. They're usually more complex than an inorganic compound. We'll take a look at some in a minute. They have carbon as the central molecule. And carbon is important because it can bind to 4 other atoms. They're held together mostly by covalent bonds, usually from the carbon being bonded to other carbons, holding the molecule together. Inorganic compounds typically lack carbon, but inorganic compounds are required by living organisms for the organism to grow, to repair themselves, to maintain themselves, and for reproduction. Examples of some inorganic compounds include water, which is actually the major molecule in all living things, water, H2O, carbon dioxide, and oxygen. They're all examples of inorganic compounds. And you'll note that carbon dioxide does contain carbon, so inorganic compounds can contain carbon, but they don't have to. And typically they don't, like water and oxygen don't contain carbon. Water, as I mentioned, is one of the most important and abundant molecules in all living cells. All right, so this is the start of our worksheet, so I'm not going to go over this, but you can go over the slide and it'll help you with the worksheet on the molecule water, which is an inorganic compound. And let's move this over and go to the end of the worksheet. So looking at organic compounds, here are some organic compounds. Don't memorize any of them. I just want to show you they can be simple, like acetic acid. If anyone can read that, I can't even read it with my glasses. They can be more complex like this. They typically are held together by carbons linking together. Although if you notice, let's see if I can blow that up. Right there, this is held together right here by oxygen. So it doesn't have to be held together by carbon, but most of them are held together by carbon. And they can be linear structures like this one here. Let me blow that. Oops, too far. Like this one here. But they can be circular, like these here. And in this structure, wherever we have a carbon, we have a point. Okay? So wherever there's a point, there's a carbon. And then that's the carbon holding it together from here to there by a bond, a covalent bond. It should be similar to that. And when you draw it this way, you don't have to put in the hydrogens. So if there's no molecule, let me see if I can blow that up a little more. Actually, there are no molecules attached to any of these except for hydrogen. You don't have to draw in the hydrogens when you draw it this way. Any question about that? Just showing you some of the organic compounds. Methane is the simplest organic compound, CH4. And don't ask me what the most complex organic compound is. It's probably one of the biological molecules, but I don't know. All right, organic compounds always contain carbon and hydrogen, always. They may contain oxygen, like amino acids, carbohydrates, nucleotides, lipids. All of them contain oxygen, as well as carbon and hydrogen. The hydrocarbons only contain carbon and hydrogen. So the hydrocarbons are the simplest organic compounds, and hydrocarbons are like things you burn for fuel. Let's come back here. Methane, ethane, propane are all hydrocarbons. They only contain carbon and hydrogen. Organic compounds may contain other elements like nitrogen, sulfur, phosphorus, and they're all held together covalently by covalent bonds. Most of the organic compounds, if they have nitrogen, sulfur, or phosphorus, these are in small numbers, meaning carbon and hydrogen are the main, and maybe oxygen is much more common. But like, let's see, organic acids and amino acids, that's what I'm trying to say. You can find nitrogen in amino acids, that's an organic compound. Phosphate or phosphorus you can find in DNA, that's an organic compound. Compared to inorganic compounds, the organic compounds are much more complex. And the organic compounds are capable of more complicated biological functions. What are some examples of an organic chemical compound? You don't need to answer that, I've got it in the next slide. Let me shrink that. Methanol and methane are organic chemical compounds. Methanol is similar to methane. It's CH3 with an OH attached to it. The carbon of the organic compounds are often found bonded together in ring chain or chain structures like isopropanol shows you. The molecule is held together because the carbons are bonded together, holding the molecule together. And so we call this the backbone of the molecule. So the carbons typically are the backbone of the molecule. Now when we come back to this molecule here, it is true that the carbons are most of the backbone of the molecule, but the oxygen does hold it together right there. So the oxygen is part of the backbone also. This is a chain structure, and I think I mentioned this is the ring structure. Organic compounds can have functional groups bound to a carbon. A functional group is some chemical structure that gives this molecule different properties than the molecule without that functional group. Like methane, for example, here is methane. It's a gas, it does not dissolve in water, and that's just the properties that methane has. I'm sure there's other properties you could have that you could say. Oops. Methanol, on the other hand, is a liquid, I think, and it can dissolve in water, and what happens is why it's different is because of this functional group. The OH group gives this molecule properties which differ from the molecule without that functional group. Any question about that? So functional groups are responsible for most of the chemical and the physical properties of a particular organic molecule, particularly of the biological molecules, but of any organic compound or organic molecule. The functional groups give most of the chemical and physical properties. And we mentioned that the hydroxyl group, the OH, in alcohols, changes the property of the molecule, making the alcohol, methanol, hydrophilic, so that molecule will be attracted to water and will dissolve in water. Methane, as you remember, is not attracted to water and does not dissolve in water. Let's look at some of the functional groups. This table is showing you 5 functional groups. We've already talked about the OH group, called an alcohol. You do need to note the groups which are surrounded in the red boxes. This group here, the NH2 group, is called an amino group, and it is found in proteins and obviously amino acids. Okay? You should also know the carboxyl group, the COOH group, found in organic acids, lipids, and proteins. You can also find it in amino acids. And then the functional group, the phosphate, the PO4, with the negative charge. It's a phosphate group, and it's found in DNA and in ATP. It's also found in a molecule we'll talk about later, phospholipids. I don't think we'll get to that molecule today. We'll get to it on Thursday. So the functional groups, there may be more than one functional group in a single molecule. Like the amino acids, shown here, have a functional group, the amino group, the NH2 over here, and the carboxyl group, the COOH group over here. Why do you think I want you to know the groups that are surrounded by the red boxes?
Probably going to be on the test.
Well, yes, that is true, but there's another reason.
Does they have to do with like dehydration and like hydrolysis reactions for like living environments?
You're getting on it, and that is, these are the groups that you find in the biological molecules. And so we're going to be running into carboxyl groups and phosphate groups, OH groups, and amino groups whenever we talk about the biological molecules. So that's why I want you to know the groups surrounded in a red box. Like, I'm not going to ever talk about the ester link. At least I don't think I'll ever talk about it. And I'm not going to talk about the aldehyde group either, okay? So you don't need to know those groups, but you should know the groups surrounded in the red box, because you'll see them time and time again when we're talking about the biological molecules. And yes, you do need to know them because they will be on the quizzes. That's sort of the wrong reason to know them, but if that's good enough for you, then that's good enough. All right. So we talked about organic compounds. The small organic molecules can combine into larger organic molecules, and they can combine into the largest, which we call macromolecules. Macromolecules of polymers consisting of many small repeating molecules called monomers. So for the organic compounds, we have macromolecules, which are polymers, and the polymers consist of small repeating molecules called monomers. So how you make a polymer is you bring various monomers together in a dehydration synthesis reaction going this way. It's called dehydration synthesis because you pull water out, you generate water, the OH comes off and the H from this molecule to make water, and then you get a link between this R group and that R group, creating the polymer, two R's connected together. And that's how you make organic compounds, by combining two monomers into a polymer. This is actually a dimer here. In dehydration synthesis, Another name for that is condensation reaction. And yeah, I'm afraid you need to know both of those terms. I tend to use dehydration synthesis. You can split the polymer by adding water and then going this way in a hydrolysis reaction, and you add the water and it breaks apart, and an H joins to this R group and an OH joins to that R group, and then you make your two monomers. So dehydration is the mechanism for the synthesis of macromolecules in your body, such as the making of carbohydrates, lipids, and proteins. And then when you're degrading, let's say you eat a steak, you degrade the protein in the steak, you do it by hydrolysis reaction to break the polymer in the steak, the protein, down into the monomers. Let me do mention that R here is standing for an organic group or molecule, and R can be many different things. Like if we're talking about steak here and this is the protein, we'll break it down in more than one reaction. They're all hydrolysis reactions. You'll break the steak down into amino acids, and so this R group would be really everything of the amino acid except for the hydrogen, and everything here for the amino acid except for the OH group. Any question about any of that? Have I confused anyone? All right, so this is showing you dehydration synthesis. You combine in the monomer to make the polymer, and water comes out. You combine the monomer with, this actually already is a polymer, but you're making a bigger polymer. And then you can break off this monomer by adding water in a hydrolysis reaction. So I guess I should mention hydrolysis and condensation, or dehydration synthesis. They're the reverse of each other. All right? Let me get started on carbohydrates and then we'll end this lesson. Carbohydrates include sugar, starch, and other complex carbohydrates. Carbohydrates are an important energy source for living things like you. When you want energy, you typically eat carbohydrates. You don't have to, but you typically do. We use carbohydrates for structure in our body, like structure in our cells. And we use carbohydrates when we make nucleic acids. Carbohydrates consist of carbon, hydrogen, and oxygen, but they don't consist of any random number of carbon, hydrogen, and oxygen. They have a general formula. The general formula is for every carbon, there's 2 hydrogens and 1 oxygen. So for each carbohydrate, you can abbreviate it as CH2O, put it all in brackets, and then an N for the N, the number of times this unit is repeating. So for glucose, for example, N is 6. So you put a 6 here, glucose will be 6 carbons, 12 hydrogens, and 6 oxygens. Any question about any of that? Simple sugars or strings of simple sugars can be linked together to make a larger carbohydrate, and we can classify carbohydrates into 3 major groups based on their size. There's the monomers, which are the smallest, and the monosaccharides, like glucose and fructose. There's the disaccharides, and those are 2 monosaccharides joined together. Lactose and sucrose are disaccharides. And then there's polysaccharides, which are many monomers or many monosaccharides linked together to form polysaccharides. For example, starch is a polysaccharide, which is formed together just by linking a whole bunch of glucoses together. All right, any question about any of that? If not, I'm going to stop here and give you 2 minutes early. All right? So I will see you in the lab at 6:30.